Welcome back to the deep dive. Today we are undertaking a really intense biochemical expedition. The sources you've provided are highly specialized.
They really are.
We're taking a deep look into the structural secrets of the smile, specifically the protein that is, well, the very architecture of periodontal health.
We're talking about collagen.
The most abundant protein in your body, but here in the mouth, it's something else entirely. It's a structural determinant of, epic proportions.
And this deep dive is so necessary because, understanding clinical outcomes in periodontics, whether it's stability or disease progression or even successful regeneration, it's just impossible without moving past simple histology and diving deep into the molecular factory.
Right.
Our mission today is, well, it's surgical. We have to dissect the synthesis pathway of collagen, tracing it meticulously from a simple gene transcription event all the way to the mature, tough, cross-linked fiber bundles that anchor your teeth.
And then the goal is to bridge that molecular understanding directly to the clinic.
Exactly.
Yeah.
Directly to clinical issues like periodontitis, regeneration, and even diagnostics.
Yeah.
It is fundamentally about understanding the why behind the mechanical integrity and, the resilience of the periodontium.
When you really think about it, the demands placed on the oral cavity are, they're functionally absurd.
They are.
This collagen structure has to withstand constant, repetitive, and often high-magnitude masticatory forces. You are crushing things dozens of times a minute, and at the same time, fight off a constant, aggressive microbial challenge. The periodontal collagen is not just some kind of filler. It's a dynamic, resilient biological suspension system.
That's the critical initial takeaway. You've hit it right there. The structural foundation of the tooth is not static. It's not like reinforced concrete.
No.
It is a highly dynamic, rope-like protein network, and it's been perfectly optimized by evolution. Its primary function is to efficiently convert potentially destructive, high-magnitude compressive stresses, the ones generated during chewing, into tensile strain.
And that strain is then dispersed, safely dispersed over, and it even stimulates the surrounding alveolar bone. This is a mechanism of protection and maintenance, all through mechanical load transfer.
Okay, let's unpack this structural overview then. We should focus on where collagen resides and what specific job it performs in the three key anatomical pillars of the periodontium. What makes collagen the indispensable scaffolding material here, according to our sources?
Well, collagen serves as the absolute fundamental structural determinant. It is the architectural backbone for the entire periodontium.
The whole thing.
The whole thing. It is the primary scaffolding material, and it's responsible for giving the tissues mechanical strength, maintaining their three-dimensional form, and providing resilience against all those functional forces.
So if it fails, the whole system just collapses.
If this robust framework fails, the structural connection between the tooth and the bone is fundamentally compromised. The dentition becomes incapable of handling functional loads. It literally binds the soft tissues like the gingiva and oral mucosa to the mineralized structures, the cementum and the alveolar bone. So it's a strategic load-bearing component that connects hard tissue to soft tissue. Let's look at those three pillars, starting with the soft tissue, the gingiva.
Right, the gingival connective tissue, which is often called the lamina propria, it has to be exceptionally resilient. Its job is to shield the delicate underlying structures while resisting abrasion and maintaining a tight seal around the tooth.
And it does that with a specific collagen mix.
It's characterized by a very dense collagenous network, predominantly type I collagen, but with a notably high proportion of type 3 collagen.
And that type 3 adds flexibility.
Exactly. It enhances its flexibility and ability to rebound from compression. And crucially, we see a specific organization here. The gingifibers, things like the circular, dentogingival, and transeptal groups.
And a crucial distinction here, which I think a lot of people overlook, is their attachment.
Indeed, that's a great point. The gingival fibers form a tight, fibrous cuff around the tooth margin. But, and this is key, they do not attach directly into the tooth surface itself.
So what's their job then?
Their function is to secure the gingiva firmly to the adjacent bone and cementum. This distinction is vital. They provide structural integrity for the cuff, maintaining its marginal position, which is the first line of defense against microbial penetration, but they are not the primary anchor holding the tooth in the socket.
That structural anchor job belongs to the next pillar, the periodontal ligament, or PDL, the true mechanical marvel of the system.
The PDL is absolutely a marvel of specialization. It's a very narrow band of soft connective tissue, typically about 0.2 to 0.4 millimeters wide in a healthy state, and it acts as that essential biological suspensory system.
A suspension system.
Its highly organized collagen structure allows for the microscopic physiological movements, the slight rotation and intrusion that dissipate powerful occlusal forces.
Because if it were just fused, it would break.
If the tooth were rigidly fused to the bone would fracture under repeated stress. The PDL prevents that, much like the suspension system on a car absorbs road shocks.
And the collagen here is arranged into specific groups to handle those directional forces, right?
Exactly. We have the principal fibers arranged into distinct groups, where the oblique fibers are by far the most abundant. These fibers run diagonally from the cementum up to the alveolar bone, and they resist the vast majority of vertical masticatory forces.
So they're taking the brunt of the force when you chew.
They are, and they ensure those forces are transmitted as tension onto the alveolar bone, which is the ideal stimulus for bone maintenance.
Finally, we have the hard tissues, alveolar bone and cementum, where the collagen matrix provides the template for mineralization.
Here, type I collagen reigns supreme. It forms the organic matrix, about 90% of the organic composition, that provides the initial scaffolding and tensile strength before it gets densely mineralized with hydroxyapatite.
And it's concentrated at the insertion points.
Heavily concentrated at the specific insertion sites where the PDL fibers terminate. These terminal embedded portions of the principal collagen bundles are what we define as Sharpie's fibers.
Sharpie's fibers are essentially the biological anchor bolts.
They are. That's a perfect analogy. They penetrate the mineralized matrices of the cementum on one side and the alveolar bone, specifically bundle bone, on the other.
And they get mineralized themselves.
At these points, the collagen itself becomes partially mineralized, creating these incredibly firm anchoring points that secure the tooth, while still allowing that crucial micromovement. This type I collagen framework provides the necessary tensile strength that protects the inherently brittle mineralized structure from fracturing under functional stress.
This all sounds incredibly robust, but you keep emphasizing that the entire system is dynamic, that it's in a state of flux. This brings us to the core concept of health, the critical balance between synthesis and degradation.
This homeostasis is perhaps the single most important concept in periodontal health. The PDL exhibits one of the highest collagen turnover rates in the entire body. Our source cites the capability for remodeling within a stunning 1 to 6 days.
1 to 6 days. That's incredible.
It is. This dynamic capability is not a flaw. It's essential for adaptation, whether it's minor physiological drift, responding to occlusal changes, or major remodeling during, say, therapeutic tooth movement.
How is this equilibrium maintained at the cellular level? What's actually happening?
Homeostasis requires periodontal fibroblasts to be hypervigilant and constantly active. They have to simultaneously synthesize vast amounts of new, pristine collagen fibers, while also secreting precise amounts of enzymes, specifically the matrix metalloproteinases, or MMPs, to degrade and remove the old, stressed, or damaged fibers.
So production has to equal removal.
Precisely. It ensures that production precisely equals removal. When that delicate equilibrium shifts, when demolition outpaces construction, that is the biochemical linchpin of disease initiation.
Now we need to zoom right in and examine the raw material itself. To appreciate the stability of the final collagen fiber, we have to look at its unusual molecular blueprint. The source highlights this unique amino acid code that makes up collagen.
Collagen biosynthesis starts with a very demanding structural requirement. For the final triple helix structure to form, the primary amino acid sequence must follow a precise pattern. We see an unusual composition where glycine, proline, and its derivative hydroxyproline collectively account for roughly 50% of all residues.
Wow, half of the whole protein is just 3 amino acids.
And this is dictated by the repeating sequence, glycine XY.
And the requirement for glycine at every third position is absolute. What happens if there's a genetic mutation, even a minor one, that substitutes a larger amino acid there?
That's a fantastic question that really probes the absolute limits of the molecule. Glycine, or galorenti, must occur at every third position because it is the smallest possible amino acid. It completely lacks a side chain.
It's just a hydrogen atom.
Just a hydrogen. If a residue with any size, even alanine, which is tiny, were present at that crowded central axis of the triple helix, the 3 polypeptide chains physically could not pack closely enough together.
They'd bump into each other.
The resulting steric hindrance would prevent the tight, rod-like conformation, and the helix would fail to form correctly. Glycine is the zero-clearance component that allows the chains to braid tightly together.
So glycine is the space requirement. What role do proline and hydroxyproline play in setting the geometry of the chain? They must be just as important.
Oh, absolutely. Proline, or pro, usually occupies the X position. Its chemical structure is unique. It forms a rigid, five-membered ring that imposes fixed angles on the polypeptide backbone.
It puts a kink in the chain.
It does. This cyclic structure, in turn, enforces the extended non-coiled conformation of each individual chain, which is known as the polyproline 2 conformation. This configuration is critical because it explicitly prevents the chain from folding into an ordinary alpha helix, instead favoring the formation of the larger right-handed collagen triple helix. Proline sets the necessary twist.
And then hydroxyproline comes in to lock that twist into place.
Precisely. Hydroxyproline, or HIP, typically occupies the Y position. HIP is just proline that has been post-translationally modified by adding an OH group. That hydroxyl group is absolutely necessary because it forms crucial water bridges, hydrogen bonds mediated by water molecules, that greatly stabilize the three intertwined polypeptide chains of the triple helix.
It's like molecular glue.
It really is. Without sufficient HIP, the entire collagen structure lacks that necessary molecular glue, making it unstable and poorly folded, particularly at body temperature.
So if proline provides the twist, hydroxyproline provides the covalent and hydrogen bond glue that locks the three separate chains into one robust braided cable. Now let's trace this construction process inside the cell. Where does this intricate intracellular biosynthesis take place?
It all initiates in the nucleus, where the collagen genes, like SOL1A1, which codes for the primary alpha chain of type I collagen, are transcribed into mRNA.
Okay.
That mRNA then moves out to the rough endoplasmic reticulum, or RER, where it is translated into the nascent single polypeptide chains, the procollagen chains.
This is where the magic and the absolutely critical stability-determining modifications happen before the chains even have a chance to leave the cell.
Yes. The post-translational modification step inside the RER is paramount for generating a stable fiber. Inside the RER, the nascent chains undergo massive hydroxylation and subsequent glycosylation.
This is where the proline becomes hydroxyproline.
This is where the specific proline and lysine residues are modified by the addition of hydroxyl groups, forming hydroxyproline and hydroxylysine. It's an enzyme-driven assembly line. Once sufficient hydroxylation occurs, the three chains self-associate and begin to wind up into the triple helix conformation.
And that stable triple helix is then moved along the cellular processing pathway.
Correct. The correctly folded procollagen, now a stable triple helix thanks to those modifications, is transported from the RER to the Golgi apparatus for final modifications, packaging into large secretory vesicles, and eventual transport to the cell surface.
And this specific enzyme-dependent post-translational step is the exact point where a common nutritional deficiency can lead to catastrophic structural failure, which is the famous vitamin C connection.
This is perhaps the most vivid example in biochemistry of a micronutrient deficiency causing a macro-level pathological collapse. Vitamin C, or L-ascorbic acid, is not just helpful, it is an absolutely essential cofactor for two key enzymes: prolyhydroxylase and lysyl hydroxylase.
And these are the enzymes responsible for creating the HIP and HYL that stabilizes the helix.
Precisely. Vitamin C has to be present to keep the iron atoms in the active site of those enzymes in the reduced state, which is FAOF. Without sufficient vitamin C, the enzymes rapidly become inert.
They just stop working.
They stop working, and this leads to the production of severely underhydroxylated collagen.
Let's visualize that failure for the listener. What exactly happens to the triple helix without those hydroxyl groups?
Without sufficient hydroxyproline, the chain structure is compromised in two major ways. First, it lacks the necessary rigidity provided by the locked geometry. Second, and most critically, it cannot form the crucial water-mediated hydrogen bonds that tie the three strands together.
So the glue is missing.
The glue is missing. The underhydroxylated collagen is thermally unstable. Its melting temperature, or denaturation temperature, drops from a stable 42 degrees Celsius down to around 24 degrees Celsius.
Wait, 24 degrees Celsius is just room temperature?
Exactly. At normal body temperature, 37 degrees Celsius, the collagen quite literally denatures. It unravels because it cannot maintain its triple helix structure. The rope holding the periodontal ligament together just disintegrates.
And the clinical consequence of that molecular unraveling is scurvy.
Yes. That structural failure manifests clinically as scurvy because the fundamental collagen structure is defective throughout the body. You see capillary fragility, generalized bleeding, easy bruising, and locally in the mouth, fragile, bleeding gums, weakened periodontal attachment, and eventual loss of teeth due to defective alveolar bone and PDL collagen formation. It is a systemic structural collapse rooted in a molecular failure to stabilize the triple helix.
Wow. So once the stable folded procollagen leaves the cell via exocytosis, what happens in the extracellular space to transform that soluble molecule into a rigid, load-bearing fiber bundle?
This is the maturation phase. The first step involves enzymatic cleavage. The procollagen molecule possesses large terminal propeptides at both the N and C ends.
The safety locks we mentioned earlier.
The safety locks. They were essential inside the cell to prevent premature assembly.
Right.
Now, they must be removed by specific enzymes, procollagen N proteinase and C proteinase. Once cleaved, the molecule is now called tropocollagen.
That system keeping it soluble until it's outside is elegant biochemical quality control.
Absolutely.
Yeah.
Once it's tropocollagen, it spontaneously and precisely aligns itself in a quarter-staggered pattern. This means the tropocollagen molecules overlap by exactly 3/4 of their length.
And that creates a visual pattern.
It does. This specific repeating pattern is what creates the characteristic 67-nanometer banded febrile appearance you see under an electron microscope. It's beautiful, really.
And the final step is strengthening the assembly, making it ready to bear the immense tensile load it will face in the PDL.
That final crucial step is covalent cross-linking. This is irreversible and provides the ultimate mechanical strength and resistance to subsequent enzymatic degradation. This process is catalyzed by the enzyme lysyl oxidase.
What does lysyl oxidase do?
It oxidizes specific lysine and hydroxylysine residues on adjacent tropocollagen molecules, creating strong covalent bonds between the chains.
So if the triple helix is the braided rope, cross-linking is welding those ropes together side by side to form a strong, resistant cable.
Exactly. A perfect way to put it. If this step is inhibited, and lysyl oxidase is copper-dependent, so a copper deficiency can inhibit it, the fibers remain weak and susceptible to structural failure, even if the initial helix folding was successful. Cross-linking defines the mechanical durability of the final periodontium.
We've established how the fiber is built, from glycine to the final crosslink. Now we can examine how these fibers, specifically type 1, type 3, and the minor but crucial type V, are specialized to make the periodontal ligament function as that critical, resilient shock absorber system.
The PDL is, well, it's a masterpiece of composite material science. It needs to be incredibly strong to prevent tooth intrusion, but also highly elastic to manage and dissipate shock. The different collagen types mediate this balance.
Let's start with the dominant player, type I, which we can think of as the structural steel of the system.
Type I collagen is absolutely the structural backbone. It forms thick, densely packed fiber bundles that provide the principal tensile strength and stiffness, offering maximum resistance to stretch and heavy load. It is the dominant component of the bone, the cementum, and the thick, load-bearing principal fiber bundles within the PDL. Its role is sheer mechanical resistance.
And type 3, I understand, introduces flexibility into the system. It's the softer component.
Type 3 is the elastic mesh, or the compliance component. It forms finer reticular fibers. Critically, type 3 is highly abundant in the PDL and gingiva compared to other dense connective tissues.
So it's a special feature of this area.
It is. It is co-distributed with type 1, and its presence confers elasticity and flexibility, which are essential attributes for rapid shock absorption and allowing the tissue to rebound quickly after the cessation of masticatory force.
Interesting.
And what's more, type 3 is highly upregulated during the initial phases of wound healing. It provides a pliable, temporary scaffold before it's eventually replaced by the stronger, stiffer type I fibers.
So we have strength from type I and elasticity from type 3. Where does type V fit in? The source notes it's crucial despite its minor presence.
Type V collagen is the quality control manager, you could say, or the fibril nucleator. While it accounts for only a small percentage, it is indispensable because it controls the organization and the diameter of the much more abundant type I fibers.
How does it do that?
Type V co-localizes with type I and is believed to form the core of the heterotypic fibrils. By regulating the overall diameter and spacing of the massive type I fibers, type V ensures the final fiber network is homogeneous, has the correct structural integrity, and maximizes the tissue's biomechanical performance.
That structural hierarchy strength, elasticity, and controlled diameter is what allows the PDL to earn the name biological suspensory system. So how exactly does this network manage those massive occlusal forces without causing microtrauma?
This is the essence of mechanotransduction in the PDL. When you apply high-magnitude compressive occlusal forces, the organized PDL network acts as a pressure vessel, converting that compression into tensile stress.
It changes the direction of the force.
Precisely. The orientation of the oblique fibers ensures that this tensile stress is transmitted safely and broadly onto the alveolar bone surface. This load transmission is protective. It prevents the root from being jammed directly into the bone, and perhaps more significantly, the tension provides a positive mechanical stimulus that actively promotes bone preservation and maintenance. It prevents disuse atrophy.
This implies a dynamic adaptation that must be incredibly rapid, given the forces we exert daily.
It is remarkably dynamic. As we noted, the turnover rate is astonishingly high, 1 to 6 days. This high turnover is directly governed by mechanotransduction, which is the cellular ability of PDL fibroblasts to sense and interpret mechanical strain.
The cells can feel the stretch.
They can. These fibroblasts are equipped with integrins and other mechanoreceptors that read the physical stress, essentially reading the load like a text message. If the message indicates tension or strain, it activates specific intracellular signaling pathways that increase collagen gene transcription COL1A1 and COL3A1. This allows the PDL to constantly reorganize, thicken, or thin in response to minute shifts in functional demands.
And the most dramatic clinical expression of this dynamic adaptability is controlled orthodontic tooth movement.
Absolutely. Orthodontics is nothing more than the carefully controlled manipulation of PDL mechanobiology. When you apply a sustained force to move a tooth, the PDL responds predictably. On the pressure side, where the PDL fibers are compressed,
They get squished.
and this strain induces the release of inflammatory mediators, which stimulates osteoclast differentiation and activity. The result is targeted bone resorption, allowing the tooth to move.
And on the opposing side, where the PDL is stretched,
On the tension side, the stretching of the fiber bundles signals the PDL fibroblasts to synthesize new collagen and activates osteoblasts, the bone-forming cells, to deposit new bone matrix. This coordinated destruction and deposition allows the tooth to migrate through the bone, critically maintaining its functional attachment at all times. This whole process is mediated by the integrity and adaptability of the collagen matrix.
and the physical connection of this dynamic PDL to the rigid mineralized tissues is mediated by Sharpie's fibers. Let's delve a bit deeper into what makes these anchors so effective.
Sharpie's fibers are the terminal anchors, comprised of the embedded portions of the principal collagen bundles, which are predominantly type I. They function as robust transitional interfaces.
So they're part organic, part mineral.
That's a great way to describe it. They originate as fully organic viscoelastic PDL fibers, but as they penetrate the mineralized matrices of the cementum and the adjacent alveolar bone, they become partially mineralized. They essentially knit the PDL directly into the hard tissue.
That partial mineralization sounds like a very sophisticated design choice.
It is. It provides exceptional resistance to pull-out forces or tensile failure while minimizing stress concentration points, thereby securing the tooth firmly within the socket. The strategic organization of the entire fiber system, the oblique fibers resisting vertical pull, the horizontal fibers stabilizing laterally, all anchored by the mineralized Sharpie's fibers, ensures maximum load handling capability while preserving the necessary elasticity for functional movement.
We've spent considerable time detailing the incredible balance required for periodontal health. Here's where the conversation shifts to pathology. What happens at the molecular level when that exquisite homeostasis collapses, initiating periodontitis? This is the war in the tissue.
The shift from health to disease is characterized by a dramatic molecular upheaval where matrix synthesis effectively halts and enzymatic degradation accelerates catastrophically. The primary trigger, as documented, is chronic inflammation and a sustained bacterial challenge, specifically lipopolysaccharide, or LPS, from keystone pathogens like P. gingivalis.
And this triggers an immune response.
An immediate and overwhelming immune response.
And the immune cells, which are there to protect, ironically become the agents of structural destruction.
Precisely. Neutrophils, the first responders, flood the area and release massive concentrations of highly potent collagenases, primarily MMP8 and reactive oxygen species, or ROS. But the core mechanism of structural failure lies in the cytokine feedback loop.
The cytokines are the signaling molecules.
They are pro-inflammatory cytokines, interleukin-1 beta, tumor necrosis factor alpha, and IL-6 are massively upregulated, and they serve to completely reprogram the periodontal fibroblasts.
How does that reprogramming translate into massive, irreversible collagen loss?
The mechanism is a dual attack. First, the cytokines severely inhibit the constructive side of homeostasis. They downregulate collagen synthesis by reducing the mRNA levels of procollagen genes.
So the factory shuts down.
The factory shuts down. Fibroblasts effectively stop trying to repair the matrix. Second, and far more destructive, these same cytokines massively upregulate the secretion and activation of the matrix metalloproteinases, the MMPs.
So construction stops, and the demolition crew gets an unlimited budget, fueled by inflammation.
That analogy holds perfectly. MMPs are a family of zinc-dependent endopeptidases. They are essential for normal, controlled turnover. But their ability to cleave the rigid, triple helical structure of collagen makes them uniquely destructive.
Why are they so special?
Because native, highly cross-linked collagen is resistant to virtually all other proteases. Only MMPs can really get in there and chop it up efficiently.
What's the significance of that zinc dependency?
It's a key structural requirement. MMPs require a zinc ion in their active site to hydrolyze the peptide bonds within the collagen helix. This dependency is actually a potential therapeutic target. Chemicals that chelate or bind zinc can inhibit MMP activity. However, in periodontitis, the sheer volume of secreted MMPs, particularly those released from inflammatory cells, just overwhelms the system.
And their activity is usually controlled by TIMPs. What is the tipping point that causes the structure to collapse?
The tipping point is the severe imbalance between active MMPs and tissue inhibitors of metalloproteinases, or TIMPs. In health, TIMPs tightly regulate MMP activity. In periodontitis, MMP expression is strongly elevated and the ratio shifts dramatically. We get an excess of active MMPs and often local functional inhibition of TIMPs, leading to uncontrolled collagenolysis.
Let's identify the specific destructive agents. Which MMPs dominate the war in the periodontal tissue?
We have to highlight two. MMP8, or neutrophil collagenase, is the undisputed primary collagenase detected in high concentrations in inflamed periodontal tissues. It's released rapidly by those first responder neutrophils.
It's the first on the scene.
And it's the most reliable indicator of acute ongoing destruction. And then there's MMP13, or fibroblast collagenase 3. This one is highly potent. It's upregulated by the inflammatory cytokines and is highly effective at cleaving both type I and type 3 collagens, actively dismantling the foundational framework.
The quantitative result of this uncontrolled enzymatic activity is simply staggering.
It is irreversible destruction. This process leads to the rapid dismantling of the periodontal attachment. The sheer volume of destruction studies indicate that up to 70% of collagen can be destroyed in actively inflamed gingival areas, result in pocket deepening, loss of functional attachment, and subsequent alveolar bone height loss.
So the clinical diagnosis is just what we see after the molecular damage is done.
The clinical diagnosis of periodontitis is fundamentally the macroscopic realization of molecular collagen destruction.
Yes. We must also expand our discussion beyond local infection and consider the systemic factors. Our sources emphasize that collagen integrity is highly susceptible to overall physiological changes. You referred to it as a biological sentinel.
That description is apt because the collagen matrix reflects the patient's overall physiological and metabolic status. The source highlights that collagen metabolism is tightly regulated by systemic hormones. This is not a subtle effect, it is fundamental.
Let's look at the building influences first.
Okay. Hormones like estrogen and growth hormone, or IGF-1, are anabolic. They enhance fibroblast activity, boosting the transcription and translation of type I and type 3 collagen production, thereby strengthening connective tissue throughout the body, including the periodontium. This is why periods of hormonal increase, like early puberty, often lead to robust tissue.
Conversely, stress and specific metabolic states have a profoundly catabolic or destructive effect.
Correct. Catabolic hormones, such as cortisol, the primary stress hormone, suppress fibroblast proliferation and directly inhibit pro-collagen mRNA levels. This effectively pauses the repair process and weakens existing connective tissue. High chronic cortisol levels compromise the PDL's ability to maintain its rapid turnover, making it vulnerable to breakdown during functional stress.
And the sources also point specifically to advanced glycation end products, or AGEs, often linked to systemic conditions like diabetes. This adds another layer of systemic compromise.
AGs are critical. In uncontrolled or poorly controlled diabetes, excess glucose reacts non-enzymatically with long-lived proteins like collagen in a process called glycation. These AGs accumulate, particularly in tissues with slow turnover like bone and long-lived PDL fibers.
And what do they do to the collagen?
Structurally, AGEs introduce irreversible, random, and unwanted cross-links into the collagen matrix.
How does that unwanted cross-linking harm the function of the collagen? I thought cross-linking was good.
The purpose of biological cross-linking, the kind catalyzed by lysyl oxidase, is to add controlled strength. AGE cross-links are random and excessive, making the collagen stiff, rigid, and brittle.
So it loses its elasticity.
It does. This impairs its function as an elastic load dissipator, leading to microfractures and increased susceptibility to degradation. Furthermore, AGEs interfere with the removal of old collagen, impairing the natural healing response of the PDL. If the systemic environment is compromised by high AGE levels, the periodontal collagen framework suffers dramatically, further reinforcing that the periodontium is not an isolated structure.
Given the massive destruction caused by this MMP cytokine loop, what is the biological limit of regeneration once the inflammation is finally controlled through therapy?
Well, partial regeneration is certainly possible, but it requires two things. First, the complete cessation of chronic inflammation, and second, time.
Right.
Once the inflammatory cytokine levels drop, fibroblasts can gradually resume collagen production. Controlled, normal levels of MMP activity, now regulated by TIMPs, again, allow for matrix resynthesis.
But spontaneous full regeneration, back to the original anatomical and architectural perfection of Sharpie's fibers and PDL bundles, that's rare.
That is the crucial limitation. The microenvironment within a healed pocket is typically fibrous scar tissue. It's not conducive to restoring the complex layered original architecture of the PDL, cementum, and alveolar bone insertion sites. Therefore, restoring full biological function and structure often necessitates actively guided therapeutic interventions, which is where regenerative medicine steps in.
Understanding the molecular war is only valuable if it translates into improved clinical care. So let's look at the immediate applications, starting with a revolutionary shift in diagnostics. Using collagen degradation products as biomarkers.
This is a huge shift. Traditional periodontal diagnosis relies on macroscopic, retrospective measures, things like attachment loss and pocket depth, which are measures of cumulative damage.
They tell you what already happened.
Exactly. Biomarkers, however, offer a non-invasive molecular assessment of ongoing destruction. They detect the real-time activity of the disease at the molecular level, allowing us to intervene before the irreversible macroscopic damage is fully realized.
What are the key molecular markers of collagen destruction we can measure with now?
We focus on two main molecular players that show strong correlation with active disease. First, the enzyme itself, active MMP8, AMMP8, because it's primarily released by neutrophils, measuring its active form in gingival crevicular fluid, or GCF, or even saliva, serves as a powerful indicator of acute active tissue breakdown.
And the second marker is the actual recognizable fragment of the destroyed collagen itself.
That's ICTP, the carboxy-terminal telepeptide of type I collagen. ICTP is a specific, stable fragment released when type I collagen, the primary matrix of bone and cementum, is degraded.
So you're literally measuring the debris from the battlefield.
That's it. And the sources illustrate a profound diagnostic utility. ICTP levels in GCF jump exponentially from healthy sites around 1 picogram per site to periodontitis sites upward of 30 picograms per site. It directly parallels the progression and severity of bone resorption.
The clinical promise is clear. Rapid chairside detection of these markers.
Precisely. This allows for personalized intervention. If a patient shows low clinical attachment loss but high AMMP8 levels, we know they are currently experiencing rapid active molecular destruction, which demands immediate and aggressive therapy. It moves us from treating damage to managing activity.
Moving beyond diagnostics, the field of regenerative medicine is using collagen as the structural material itself. It's often called the gold standard biomaterial for periodontal regeneration. Why is collagen superior to synthetic materials?
Collagen is ideal because of its biological profile. First, biocompatibility. Being a natural protein, often from bovine or porcine sources, it has very low antigenicity and integrates seamlessly with host tissue. Second, bioactivity. Unlike an inert synthetic sheet, collagen is chemotactic for fibroblasts and even some stem cells.
It calls the repair cells to the area.
It actively promotes cell migration and proliferation into the defect space. It's not just a placeholder, it's an active participant.
And the fact that it breaks down naturally is a massive clinical advantage.
Absolutely. Biodegradability means it is broken down naturally by the host's own collagenases. This eliminates the necessity for a second surgical procedure to remove the membrane, which is a critical requirement with non-resorbable barriers like dense PTFE.
And its primary application is in guided tissue regeneration, or GTR.
Yes. In GTR, collagen membranes function as physical barriers. Their purpose is highly strategic. They prevent the fast-growing epithelial cells and gingival connective tissue cells from migrating down into the defect space surrounding the root.
It holds them back.
It does, and this is vital because it creates a protected, three-dimensional microenvironment that allows the slower-growing desired regenerative cells, the PDL fibroblasts, cementoblasts, and osteoblasts, the necessary time to repopulate the area and form a new functional attachment.
However, native collagen can degrade too quickly in the body, which defeats the purpose of the barrier, doesn't it?
That is the key technical challenge. If the membrane degrades too fast, the barrier function is lost prematurely, and the rapid-growing soft tissue invades the defect. Current solutions involve modifying the collagen scaffold structure, often through chemical or physical cross-linking techniques.
To make it tougher.
Exactly. These processes increase the material's resistance to enzymatic degradation and enhance its mechanical stiffness, ensuring the membrane maintains the defect space and provides the required structural integrity for the entire 4 to 8 weeks needed for PDL and bone regeneration.
This leads us directly into the truly exciting frontier, 3D bioprinting and precision periodontics, where we can literally design and print the perfect replacement tissue.
3D bioprinting offers a transformative method for fabricating patient-specific scaffolds using collagen-based bioinks. This technology allows for an unparalleled degree of control over scaffold architecture, moving beyond simple sheets to structures that truly mimic nature.
How does this biomimicry work in practice? What are we actually printing?
Researchers are now printing layered scaffolds designed to replicate the native periodontal complex, gingiva, PDL, cementum, and bone, in a layer-by-layer fashion. They can tailor the composition and the microscopic pore sizes of each layer.
So you could have a denser layer for bone.
A denser type I rich layer for the bone, and a looser type 3 rich layer for the PDL. The ability to control the shear stress during printing allows for the precise deposition of collagen hydrogels to create the necessary structural guidance.
And we can integrate the biological power directly into that scaffold.
Absolutely. Stem cells, specifically periodontal ligament stem cells, PDLSCs, or mesenchymal stem cells, MES, can be suspended directly within the collagen bioinks during the printing process. When the scaffold is implanted, it provides both the ideal structural template, the collagen, and the necessary regenerative cell source, the stem cells, exactly where they are needed.
Which should dramatically enhance the possibility of functional regeneration.
That's the hope.
The source also mentions the movement toward recombinant collagen. Why the desire to move away from traditional animal-derived collagen?
Well, while animal-derived collagen is effective, it presents issues of batch variability and potential, though small, risks of immune reaction or disease transmission. Recombinant collagen is engineered biotechnologically, often synthesized in host systems like yeast or bacteria, to be human identical.
So it's purer and more consistent.
It offers superior consistency, eliminates the risk of zoonotic disease, and reduces potential immunogenicity, paving the way for fully defined, highly consistent, personalized constructs that can be engineered with specific signaling sequences.
Finally, let's discuss smart biomaterials. What makes a future collagen hydrogel smart?
A smart biomaterial is designed to be responsive. Future collagen-based hydrogels or membranes are being engineered to act as sophisticated drug delivery vehicles. They can be programmed to sense a local inflammatory signal, for example, a spike in those destructive MMPs or high levels of IL-1.
And they react to that signal.
And, in response, trigger the precise, localized release of therapeutic agents.
Such as targeted growth factors or antibiotics.
Exactly. They might release antibiotics to tackle a localized flare-up or growth factors like bone morphogenetic proteins, BMPs, to accelerate bone formation, but only in the presence of inflammation, ensuring the treatment is delivered exactly when and where the tissue needs it most, minimizing systemic side effects.
Ultimately, the future relies on precision periodontics, tailoring strategies based on individual biochemical profile.
Yes, we are moving beyond the one-size-fits-all approach. Precision periodontics recognizes that individual genetic variations, such as polymorphisms in collagen-coding genes like COL1A1 or variations in the efficiency of MMP enzymes, may explain differences in the disease susceptibility and treatment response. The future demands designing regenerative and treatment strategies that are specifically tailored to an individual's unique genetic and biochemical profile to maximize the preservation and restoration of their collagen reserve.
What an extensive deep dive. We started with the molecular blueprint, the zero clearance component of glycine, saw how the lack of vitamin C causes the triple helix to unravel at body temperature, traced the dynamic life of the PDL's specialized fiber types, and examined the devastating inflammation-driven enzymatic destruction caused by MMP8. And we conclude with the incredible promise of 3D-printed, custom collagen scaffolds actively rebuilding the periodontal attachment complex.
The overarching lesson is that the structural integrity of the smile is fundamentally and inextricably tied to the health of the collagenous fiber network. Clinical success in periodontics is completely dependent on a deep appreciation of this protein, from the cell all the way to the molecule.
What stands out for you as the final takeaway, something for our listener to mull over as they digest this highly complex information?
I think it's this. We've established that collagen acts as a biological sentinel, reflecting the body's overall physiological status. We discuss stress, hormonal levels, and systemic conditions like diabetes, leading to the accumulation of detrimental advanced glycation end products.
Right.
So the provocative thought is this. Given how exquisitely sensitive periodontal collagen is to systemic health, how might future personalized dental treatments, using those chairside MMP8 or ICTP biomarkers, serve as crucial early systemic health warnings? And if the integrity of the periodontium is truly a reflection of the systemic connective tissue reserve, how closely will periodontists need to collaborate with endocrinologists and internists in the future to truly protect that essential collagen?
What a profound thought.
The oral cavity is unequivocally a window into the body's entire physiological status.
A profound thought that connects the microscopic tooth socket to the entire systemic health of the patient. Thank you for guiding us through this incredibly detailed biochemical analysis today.
My pleasure.
And thank you, the listener, for sharing these deep sources with us. We invite you to continue your deep dive into knowledge.